Okay, today we're going to focus on protein synthesis. A little bit of background. You're going to hear the word gene. What a gene is simply just a piece of DNA that is essentially a code for a particular protein. So a gene is a unit of information in DNA. It's almost like a DNA unit, almost. So we have genes for several different things that we're going to be talking about later on in our genetics unit. The central dogma of biology is the process of going from DNA to protein. Each protein is made from a gene. DNA is essentially the permanent information of life. So DNA has to be made into RNA. RNA will then leave the nucleus to get into the cytoplasm. And in the cytoplasm, we have protein being built up. So RNA essentially is a copy of that message. Now we don't want the entire DNA copied, we just want a segment of it. And we're going to be talking about two different processes going from DNA to protein. One is called transcription, and the other is called translation. So a little review, RNA versus DNA. First off, they have two different types of sugars, ribose versus deoxyribose. One is single-stranded versus one is double-stranded. RNA single-stranded. And then there is a different base in RNA. In RNA, A bonds with U, and in DNA, A bonds with T. So we have uracil replacing thymine. So three major differences. With RNA, we have three different types. The first type is messenger RNA, commonly known as mRNA. It's called messenger because it is carrying the nucleotide sequence from the nucleus to the ribosome. This is through a process called transcription, which we are going to focus on in this video. RNA is simply a copy of the DNA code. We're not replicating DNA. We're simply taking a segment of that DNA and making a copy of it, getting that code out to make a particular protein. And the mRNA is going to be in your nitrogen-based sequence. So, for instance, this might be like A, U, G, C, C, G, A, C, U, A, C, whatever. Doesn't really matter. But the key thing is every three of these letters is going to represent and end up being translated into an amino acid. Oh, there's kind of three, and we have two left over. So every three. So what we're going to find is AUG ends up being, later on, an amino acid called methionine. So we work off a three-letter sequence called codons. Second type, transfer RNA. In order for your messenger RNA to be translated, we have tRNA, which is this structure right here. It will connect to messenger RNA. Just think of your complementary base pairings. And it brings with it a particular amino acid that it'll drop off. Now, the tRNA is going to be in a process called translation. Going from the RNA to the protein code. Its codon right here is called an anticodon. It is the one that will bond with the codon of the mRNA and make sure that it's reading the message correctly, translating it correctly. If we looked at this picture, as we see, we have several tRNAs which bring in each individual amino acid to eventually be built up into a nice chain, polypeptide. Our final RNA is simply ribosomal RNA. Just think ribosome here. Ribosome, what does the ribosome do? It is the protein builder. So this is also going to be part of translation. Because in translation, we are going from mRNA to protein, specifically within the cytoplasm. So building those proteins, making up the RNA, making up the ribosome to essentially translate the mRNA into a protein language. So we have our two steps. By and large, our first step is we need to go from DNA to mRNA. And this is happening in the nucleus. Once outside the nucleus, we have mRNA being translated into a protein or a polypeptide. And this happens in the cytoplasm, specifically your ribosome constructing it in the cytoplasm. So what's important about proteins? Well, proteins are involved in a lot of different things. Hair and nails, blood, brains and nerves, enzymes, cellular messengers, antibodies, a lot of important things in your body. In terms of what do proteins do for you? Let's just focus on transcription now. So, transcription. Once again, just think of we are going from DNA to RNA. We are only targeting a specific sequence of that DNA, a gene, a segment of DNA to be copied and get that message out into the cytoplasm. That message is your RNA. So we have a pretty similar process to replication, just in a different way, since we're only copying one particular segment. Now, why is this even necessary? Well, DNA can't leave. It's too big. Why? Because it has two strands. It cannot fit through the nuclear pores. RNA can fit. We make a copy. Why can RNA fit? Because RNA, remember, only has one strand. Also, we need to protect DNA. We need to just keep it in the nucleus and just kind of do its job as storing the genetic information, and we'll make copies of what we need. Where does this happen? Well, think about where is DNA? DNA is in the nucleus. So that is where transcription is going to happen. If it's too big to leave the nucleus, this is the only place it can happen. And then when transcription is complete, the RNA will leave the nucleus through the nuclear pores and move into the cytoplasm and wait for translation to begin. The ribosome will find it. So how does this happen? This is going to sound similar to kind of DNA replication. DNA unzips. How does it unzip? Because we have our good old friend helicase to do so. It unzips at a particular location for that segment of DNA. So what's going to happen is all these hydrogen bonds are going to be broken, and we have our two strands separate at that particular gene. Step 2, we have polymerase. Remember, a builder. But in this case, it's called RNA polymerase because we are building RNA this time, not DNA. So it is going to pair up your RNA bases, complementary base pairing rules. Remember, RNA has U, so we no longer have T's when we are building our RNA. RNA is single-stranded for this very reason, because we have our template strand that is DNA, And we have our new strand, our copy, which is RNA. So RNA being formed, just this particular sequence. What's going to happen then is RNA is going to separate itself from DNA, and the two DNA strands come back together. And then this is then going to go to the cytoplasm. Carry the message out of the nucleus. Now, RNA does have some processing. Just as we talked about the importance of semi-conservative replication with DNA replication, DNA does have kind of built-in nitrogen-based sequences that almost act as mutation protectors. These are called introns. These do not code for anything. Your exons are what are going to be coded for the protein. Why this is important is if there is a mutation that occurs, your genes are simply not back to back to back with each other. So if there's a shift, which we'll talk about kind of later on, an insertion or deletion of bases, it doesn't shift everything. So it's almost like playing Battleship. Is you have all of this kind of surrounded, and you have your ships in different locations, similar to your genes. Your genes are in different locations instead of being right next to each other to protect against mutations. So your exons are what's being coded out. Your introns are going to be cut out eventually, which we term splice. Spliced out. And as they are getting kind of prepped, we do have kind of caps and tails to kind of make it an official kind of mRNA. The key thing here is understanding more your introns and your exons. Your exons is what's going to be coded. Your introns will eventually be cut out. And we move to the cytoplasm. And in the cytoplasm, this is where translation will occur, which we'll talk about later. So once again, just another visual. We have DNA kind of split. We are only focusing on this part of DNA. One segment, one gene. Polymerase is your builder, specifically of RNA, putting those RNA bases. Remember, RNA has U. Easy thing to forget. So what are the results? Is we simply have that RNA sequence. So practice. Find the complementary RNA base pairs. Well, based off of our base pairing rules, C bonds with G, A bonds with T, A bonds with T, C bonds with G. Same thing here. And then A bonds with U. Remember, we are making RNA. Kind of double checking. Same thing. We have our base pairing, and we still have that sugar phosphate backbone, which I guess I should have drawn a little bit. Here's our sugar phosphate backbone. Single stranded. A little bit different, doing kind of same general thing. Complementary RNA, kind of going around. A will bond with U, C bonds with G, A bonds with T, C and G, A and T, C and G, A with T, A bonds to U, or specific RNA, and then a lot of repetition with these types of things. And then of course, we have our sugar phosphate backbone to go with our nitrogen bases. RNA is still made up of a nucleotide. Remember your nucleotide, phosphate, sugar, base. Same general thing in regards to checking your work. Single-stranded. So the final thing, kind of looking forward, DNA to RNA, this is occurring in the nucleus. Get the message out. DNA cannot get the message out, plus we have to protect it. Translation, this will be occurring in the cytoplasm with the help of your ribosome, because the whole point is to make your protein. So DNA to RNA to protein, and we call this process the central dogma. Making protein. Okay, that marks the end of today's video. If you have any questions, please let me know. The next one will focus on the translation process of going from RNA to protein. Thanks for watching.